Okay, now coming to the magnification and resolution of both light and electron microscope. See, magnification means how much time it can multiply the image, right? Okay, and resolution means how clearly that is done. Understood? So something can have a higher magnification, but it may have a lower resolution as well, right? So when the resolution is low, your image will not be very clear. It's like your phone images only, right? When you see your phone images, you must have seen some resolution is mentioned on the in the image, right?
Yes,
Okay.
sir.
Yeah.
So what is the resolution of your, like, the images that we take? Let's say if it is an HD image, right? So what is the resolution?
Well, depends. Is it full HD or HD? Because
10,
it's HD, it's 720p, but if it's
no,
full
HD
HD,
is,
1020p.
yeah, 1080, no?
Yeah.
1080 is full
Okay.
HD.
Yeah, so you said full HD, you said 1020.
So did you say full HD or did you say HD?
I said HD,
HD 720p then.
720p.
right? So 720p is what the pixels that are there, right? Okay. Clear. So the more you increase the resolution, what happens? The better image
The
clarity
image
you get,
gets,
right? Okay, so better the resolution, better magnification, and better image clarity you will get. So when we compare the two microscopes, means when we compare the light and the electron microscopes, we see that electron microscopes have a better magnification as well as they have a better resolution as well. Clear? So if you have to define magnification, it is very simple, that how many times we can enlarge the image, right? Clear? So it is how many times bigger the image of a specimen can be observed. Okay, clear? This is what magnification is. Now, when we talk about light microscope, specifically compound microscope, so there are two types of lens, an eyepiece lens and an objective lens, right? Clear? Now, where do we see the eyepiece lens? The eyepiece lens is the one which is there on the top, right? The through which you see the object, that is your eyepiece, and the one, and objective lens is the lens that is above the specimen. Okay, clear? So there are two lenses over here. Now objective also can be changed with something like there are 10x, 15, 20. There are different types of objective lenses. Similarly, that eyepiece lens can also be changed. Okay, clear? So by combining the magnification of both objective and eyepiece lens, we will get the final magnification in light microscope, right? So to
yes.
calculate total magnification, what we need to do, the magnification of eyepiece lens and the objective lens are multiplied together. So if let's say that objective is 20 times and eyepiece is 10 times. So final resolution, final magnification will be 20 into
200.
100, right? So 200 times larger image we will observe.
Yes, sir.
Understood? Any doubt?
No.
No.
Cool. Okay, now let's come to resolution. As we learn what is resolution, that how clearly we can distinguish between two different points, right? Okay, so
Yeah.
resolution or resolving power is the ability to distinguish between two separate points. Clear? So if
Yeah.
two
Yes.
separate points cannot be resolved, right? So the image that we will get will be blurry image, right? Okay, because you are unable to clearly distinguish between two different or separate objects. Okay, clear? So if the resolution is less, what will happen? We will get a unclear image or a blurry image. Understood? Clear?
Yes.
So remember that resolution plays a very important role, that how much magnification will be there. So resolution of a microscope limits the magnification. Okay. So better resolution you will have, the better magnification you will get. Okay. Clear?
Yeah.
Cool. So here we have a line mentioned over here that there is no point in magnifying an image at low resolution, as this will just result in a big blur rather than a small blur. Okay. It's very
Okay.
simple. You click a photo in your phone. What you do is you just try to start zooming it. The more you will zoom it, what will happen?
It will get blurrier.
Blurry.
Right. It will start getting more and more
Blurrier.
blurry because you are increasing the magnification, but the resolution is limited over there, right?
Mm-hmm.
Yeah.
Okay. So what decides or limits this resolution? If you talk about, say, a light microscope, so in light microscopes, what happens is there is a wavelength of visible light, right? In light microscope, we are using which light? Visible light, right? And what is the
Yes.
wavelength of visible light?
Oh,
Um,
90-ish?
For
I'm not too
visible
sure.
light, what is the wavelength?
I don't think we did this.
400 nanometer to 700 nanometer.
Yeah, we haven't done
Okay.
this.
How much?
Yeah.
400 to 700
400
nanometers.
nanometer to 700 nanometer. This is the range of your visible light. Okay, clear? If
Yeah.
anything is before 400 means less than 400, okay, clear, that is your ultraviolet. That
Okay.
is what? Ultraviolet.
Ultraviolet.
Something that is larger than 700 is your infrared, IR. Got it?
Yes, sir.
Yes.
Remember this thing. So visible is 400 to 700. But when we talk about electron beams, electrons are very small, you all know, right? Okay.
Yes, sir.
Yep.
So the wavelength of electron beams is very, very less, and that is why they are able to produce a better image, or they have a higher resolution. Clear? So the wavelength of your visible light is too long to allow for higher resolution. Remember,
Yes, sir.
what is the difference,
Yes.
or what is the limiting factor? It is the size of the wavelength of light. Okay, for visible light, in the case of light microscope, it is 400 to 700 nanometer. But in the case of electron microscopes, it is very, very less, because here we don't use light. What we are using? We are using electrons. Okay.
Yeah.
So one
Yeah.
such example is given over here, the phospholipid bilayer, which is present in your cell membrane, right? Okay.
Yeah.
So the width of the bilipid layer is about how much time? It is around 10 raised to the power 9, sorry, 10 nanometer. Okay. The maximum resolution of a light microscope is how much? 200 nanometer. So
Yeah.
can you see a lipid bilayer with the help of your light microscope?
No.
No. You
Most
cannot
probably
see
not.
anything smaller than 200 nanometer with the help of light microscope,
Yeah.
right? Okay. So you may be able to see just a line, but you cannot study that thing. So any point that are separated by a distance less than 200 nanometer cannot be resolved by a light microscope.
Sir,
Okay. Yes, dear.
is electron microscope 0.5 or 5?
0.5, dear.
Okay.
Okay, done. Okay, now let's come to electron microscope. So we know that electron microscopes, they have a higher resolution. Why? Because the wavelength that they are using over here is very small, right? So electrons have a very smaller wavelength compared to visible light, right? So we'll see an image over here. In the case of visible light, what is the wavelength? 400 to 700 nanometer. But when it comes to electrons, electrons have a wavelength of nearly less
1
than or
nanometer.
equal to 1 nanometer, right? Okay, so more and more light can penetrate through the specimen object, and we can get a better image. Understood?
Yeah.
Okay, now coming to staining specimen. As I said that in order to study the objects under the light microscope, what you need to do, you need to stain them or color them, right? Basically what happens is everything is what is a chemical, right? When you talk about a cell, so cell has cell membrane, it has cell organelles, right? Okay, clear. So these are what? These are all chemical structures, right? Okay. So in order to see them, what we do is we color them with a specific kind of chemicals. So the stains that we use over here are nothing but specific chemicals that are taken up by the specific cell organelles. Okay, and then only we can visualize any image under the light microscope. I have already told you that for electron microscopes we don't need staining. Okay, although stains are used but not to color them, instead to improve the resolution of the image. Okay, so specimen that need to be viewed under a light microscope sometimes needs to be stained, right? Okay, so the colors that we see, right, whatever colors that we see in a microscope, it is not the actual color of the cell organelle, except in few cases, like for example chloroplast. When we see chloroplast under a microscope, they are green, right, and their actual color is also green
Green.
only, right? But for
Yeah.
other cell organelles, we get some other kind of colors. Okay, clear. So some examples are given over here. Just remember the name of those examples, right? So hemotoxylin, it is used to stain plant and animal cells nuclei, and what color it gives? Purple, brown, or blue color. Then methylene blue, it is used to stain animal cells nuclei, and it gives which type of color? Blue color.
Yeah.
Acetocaramine, it stains chromosomes in dividing nuclei of plant and animal cells. Whenever we study meiosis or mitosis, means the cell division, right? So in order to study cell division, which type of stain is used? Acetocaramine, remember. Okay.
Caramel, yeah.
Then iodine, we know iodine is used to stain starch containing materials. Okay. And
Yeah.
which color it will give?
Black. Blue black.
Blue
Blue
black.
black.
Okay. Then toluidine, it again it is a blue color. It is a stain that is used to sorry, this chemical is used to stain those tissues which have DNA and RNA, and the color that they will give is blue color. Then fluoroglucanol, it is a chemical that is used to stain which chemical? Lignin, right? And lignin is found in dead plant cells. Which
Trees.
plant cells?
Dead plant cells.
Dead plant
So we
cells.
were learning
Okay.
about this in school. So like the negative negative cell, something like that, gram negative
That is
terms.
for bacteria here.
Oh,
Okay.
okay.
That is a bacteria, and that is a different staining method that is called gram staining. So in
Okay.
gram staining, some bacteria they take up color, so they are called gram positive, and those who don't take color, means they remain colorless, they are called gram negative bacteria.
Yeah.
Okay, clear?
Yeah.
Now, when we use these stains, right, for some tissues or for some specimens, we may have to just use single stain. With just one stain, we can do everything. But in some cases, we may need to use multiple stains. That is called differential staining. Okay. When we are using multiple stain or dyes in a single specimen, then that is called differential staining. Now, the more important thing for you is to remember the examples over here. Okay.